The broadcast is now starting. All attendees are in listen-only mode.
Good morning and hello to everyone, and thank you for joining us today. You've logged on to the joint Nelson Labs and Greenlight Guru webinar called Design Change Impact on Biocompatibility and Documentation. Now, I just want to go over a few housekeeping items before we start. If you miss any one of our webinars or would like to simply refer back to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars by liking Nelson Labs and Greenlight Guru on social media outlets, Facebook, Twitter, and LinkedIn. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know that every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. Nelson Labs partners with Greenlight Guru as the EQMS and medical device success platform of choice, as it is the only all-in-one solution that helps get your products to market faster with less risk and advance your success in market. With over 600 medical device clients across the globe, Greenlight Guru's platform streamlines your product development, documentation, and quality processes in alignment with major industry standards and regulations while achieving closed-loop quality system traceability. So let's get started. Today we're joined by Thor Rollins, Director of Toxicology and E&L Consulting, and John Speer, founder of Greenlight Guru. Now for a brief introduction, Thor is the Director of Toxicology and E&L Consulting. He's a certified microbiologist and specializes in the selection and conduct of in vitro and in vivo biocompatibility tests. He actively speaks on biocompatibility-related topics through Nelson Labs' external seminars, webinars, and at trade shows. He's a participating member on all Association for Advancement of Medical Instrumentation, TC 194, and ISO 10993 committees, and plays an active role along with the FDA and regulatory committees developing standards, discussing biocompatibility methods, and voting on changes to those standards. As one of a select group of experts in the industry, Thor's participation on the committees offers him insight on industry changes and helps prepare clients for changes in testing. John Speer has over 22 years of experience in the medical device industry, with time in quality management, product development, and project management at Cook, Inc., Theron, Inc., and Matrix, LLC. He is experienced in managing multiple projects and taking medical device concepts through development, regulatory submission, and to market. His expertise in the design and implementation of FDA-compliant quality management systems. He's also the host of the #1 most downloaded podcast in the industry, the Global Medical Device Podcast. And now to begin today's presentation, I turn the time over to Thor and John.
Thanks, Mike, and thank you everyone for attending today's presentation, be it morning, afternoon, evening, wherever you may be around the world. We're always excited to kind of meet with you and kind of discuss these types of topics. This is one that we deal with on a daily basis. So to kind of go through a quick agenda, we're going to be doing, Mike already did some introductions. We're just going to kind of feed into those. We're going to kind of talk about changes and identifying and evaluating those changes and the impact with biocompatibility. And then John's going to talk about how we effectively and correctly document those changes and then remain compliant amidst the continuous change. So that's going to be the flow today. Obviously, I'm really excited to speak with John. I've known John for a very long time. He's very respected, and I'm excited to hear what kind of add he has to this presentation. So it's a pleasure for both of us to be here today with you. So first off, I think it's important to kind of understand what constitutes a change. As we're talking about change, and whenever we deal with changes, and this has been something that's kind of been very popular recently with MDR. We have products that are on the market for a long time, and now that they have to redo their submission under MDR, they go back through the design history files and they realize that they may have made some changes, or maybe someone else made changes, and now we're dealing with the changes and impacts under a new regulatory system. So what I always tell people whenever we talk on the phone, and like I said, this happens to us here at Nelson Labs daily. We have clients that call us up with either a needed change or something that's been proposed to be changed in their approved device. We always tell them that any change needs to be evaluated. That does not mean that every change needs to go through testing or needs to even have some kind of really risk-based approach with toxicology, but every single change needs to have some kind of thought discussion written down on why that change is impactful or not, and what your steps are to mitigate any risks from that change. So that's so important. I can get into stories. We don't have a ton of time. Maybe we should cut back on our introductions and give us a little bit more time to talk. But just kidding. But there are things that we never would have thought that would have had an impact from a biocompatibility standpoint, like even just moving locations. When someone just moved a mile down the street, and then all of a sudden they found out that they're failing cytotoxicity based on their new location. So there are changes that have an impact, and we may not even realize it. So any change needs to be evaluated. And then you go into the regulatory aspect. I just have something here from the FDA as far as intent, which I think is important there to understand what the intent of this change is. But they mentioned that if it significantly affects the safety or effectiveness of the device, then you need more in-depth evaluation, including maybe a new 510K. For me, I think that intent is a big important word when you start looking at changes, and needs to be recorded on why you're making these changes and the risks to the patient, and then any steps you took to mitigate those risks if you identify it. So some of the changes we see a lot are label changes. This actually, with MDR, this has happened where people now have to start looking at CMRs potentially on labels. Maybe they're changing, and some more impactful kind of changes is maybe they're having a new intended use, or maybe they're looking to increase the amount of time that the device can be used. Maybe they're going to go into neonates or infants, where it's before it was only adults. These kind of things we have to evaluate. We have to evaluate the potential impact of these changes. Another change that we find that some people don't realize can have a big impact is packaging. The type of residuals that can come from sterilization and other processes, including packaging, that can be leached from that and put on the device, can have an impact. Now that we're doing more extractable, leachable testing on these medical devices, the more we're seeing that this has an impact. We're seeing chemicals coming from packaging and then getting onto the medical device. And so if you're changing your packaging materials or even setup, those kind of evaluations need to happen. Obviously, material process change, this is the most common. Your supplier either changes their material and they swear to you it's the same, or the supplier goes out of business, or maybe you're looking to get more suppliers, things like that, or you're changing a process and then you have to evaluate that change. Each one of these need to be evaluated on a case-to-case basis. If marketing, and by the way, I love hearing the discussions between engineers and marketing. I have a lot of friends in marketing, and I understand that the point and the process, but man, sometimes adding something or changing a color because they want to add a color or change a color can cause a lot of issues to biocompatibility or even paperwork. And so I always kind of talk about, do you have to do that? And I know that there's the internal discussion that needs to happen, but just remember, any of those changes, specifically adding to a coloring to a device, should have sufficient or substantial evaluation. And like we mentioned, location change. So these are all things that we see could have an impact on biocompatibility. So the first step to any change, and from a biocompatibility standpoint, is your initial risk assessment. We want to see if this change is impacting patient contacting skin or patient contacting portions only, Or is it only contacting intact skin, or is it going somewhere into the vascular system? Is this direct or indirect patient contacting? Obviously, if this change is impacting a portion of the device that does not have direct or indirect patient contact, then the risk assessment's easy from a biocompatibility standpoint. John is going to talk about the other side of documentation. But from biocompatibility standpoint, it's easy. If it doesn't have direct or indirect patient contact, there's no risk to the patient. So that change becomes a little bit easier to justify. The other thing that we see is, if you're changing this to significantly improve the clinical outcome, then we want to include that in the risk, benefit ratio. I talked about colors. We dealt with one that's not too long ago where the surgeon was, there's different sizes, and the surgeon was putting different sizes on the different ends and kind of getting that mixed up in the different options. So we had the company had colorants to be able to match up the right components. And even though that's adding a colorant, that really helps mitigate that risk of a clinical problem. So, those kind of things help when you start looking at why you're making that change. Or, you're mitigating a known risk to how the device is used. Are you making it stronger if it breaks, or things like that? So these are aspects that we want to include in the risk assessment. And then we also have to think about any unintended consequences of these changes. I'll give you one example. We had a customer a couple of years ago change the packaging label on their device. And actually, it was inside the sterile barrier. It was like a pamphlet or something like that. They were changing the color they used in that pamphlet. And so when the customer went through the risk assessment, says it's not patient contacting, so therefore no change evaluation is needed. But as soon as they went through the sterilization process that had some humidity present, they started realizing this ink was leaching or leaking and getting kind of potentially on the device. In fact, it was getting on the device after we did some chemistry. So we then had to evaluate that potential change, even though that was not intended to have any biological risks. We then noticed that it did have biological risks, and we had to evaluate it. So, and then if you do have risks that you identify, what testing you need to do. I can tell you most changes we deal with, we do not have to repeat all of the biocompatibility tests. There are times when there's a significant enough change or a significant risk that we do repeat all biocomp, but very often we're able to evaluate the risks, mitigate the risks of that change by selecting the appropriate test needed on that risk. So, but the first step to any change is that initial risk assessment. I faintly stole this slide from a friend of mine, Edward Vardy from Ethicon, but for me, this is a great way to kind of show what we talk about as risk, which is, from 14971, the probability of occurrence of the harm and severity of that harm. I love that little mouse talking about that helmet. Is it going to mitigate the risk of the cheese and that's or the trap? Excuse me. And that's basically what we're trying to do here, is try to evaluate what we need to do to mitigate any potential risks, and then understanding how, if the probability of that happening, and if it does, what's the severity of the two important things that we put into a risk assessment. The other thing I want to mention is this falls right in line with the FDA guidance document, which is actually a really good interpretation of ISO 10993. If you don't know this guidance document backward and forward, honestly, and you're into biocomp, this is probably something you should spend some more time on. But this paragraph is from what we consider a BEP, a biological evaluation plan. But the same concepts are there. If you're looking at changing a material or if you're changing a process, those need to be looked at based on the clinical use of the device. And then you should come up with a plan to mitigate those risks. And that could be either by evaluation or testing based on the type of risk and how you evaluate it. So the initial thoughts as you go into any biocomp assessment for a change is, like I talked about, is it directly or indirectly patient contacting? Obviously, that's going to help evaluate your first step, your first gauge in the risk. If it touches the patient directly or indirectly, then obviously you're going to have to do some risk assessment. Then you start looking at material similarities. And this is an interesting one because I can tell you tons of times I have people who say, well, I'm just changing a polypropylene to a polypropylene or polyethylene to a polyethylene. So there's no impact. You talk to any polymer scientist out there, and I can tell you that every single polypropylene is not the same. Every single silicone is not the same. There's additives, there's stabilizers, there's all these different things that they're putting into these polymers that changes changes not just their makeup and their performance, but also their biocompatibility risks. So understanding the similarities between the materials is important. Some of the easier ones, if you're changing a supplier of titanium, the ASTM grade titanium to another ASTM grade titanium, or stainless steel to another stainless steel to another supplier, those are a little easier. They're more controlled, but you still need to be evaluated, but testing is usually less. Polymers tend to have a little bit more risk associated with them, even if you're using the same type of polymer. The other thing that's important to do, and a lot of people don't think about this, but from a biocompatibility perspective, it's all about dose to the patient. So the size of the patient contacting change is very important. So if you have a very big tubing set, for example, and you're changing just a very small component of a stopcock, for example, that change to the overall biocompatibility risk of that tubing set could be pretty low. And even if you repeated all of your biocomp, when you start looking at the ratios of surface area to volume that we use to test, you could be just testing the exact same device, right? So we want to understand the patient contacting surface area change percentage, and that goes into our risk assessments. So I'll talk a little bit more about that in a second. So when you start looking at your device materials, some of the things that we always ask our customers to get in place beforehand is these, material manufacturing agreements. You should have really solid relationship with your suppliers. And I know sometimes that's harder said or easier said than done. A lot of times these suppliers are supplying other industries, and we're probably pretty small amounts when it comes to actual revenue, so they may not like it. I find it's better to put these agreements in place when they're trying to get your business than when you already have your business. But manufacturing agreements, no changes needed. Composition disclosures are super important. Hopefully you can get the compositions of the materials and also what they're using in their processing. That's the next thing, what chemicals, residuals could be there that we have to pay attention to. You can always get MSDSs or SDSs. I'm not a big fan of these. I mean, they're useful, but they're not peer reviewed, and they get bad information sometimes. And then some people have device master files with the FDA, which are also useful. Now, the one interesting thing, skin contact. So does your change only impact skin contacting portion of the device? If it does, the FDA has a new draft guidance out about skin contacting materials. Now, I wanted to emphasize this is draft. So, the FDA doesn't necessarily have to recognize this yet, but it does talk about their current thinking, their current thought process when it comes to risks. And they divide this up into approved materials that contact the skin, so basically low-risk materials, and then kind of non-approved or the materials that you can't just kind of say, oh, it's on the list. I call it the nice and naughty list. There's a webinar I gave on this. If you guys are interested, you go to the Nelson Labs education page. But basically, these are the nice list materials. So if you're changing a portion of your device that contacts skin only, and if it's one of these materials, then you can kind of take the same kind of concept where these materials are considered low risk, especially for skin contacting. We contact these materials all day long, every day with our skin. And so, not saying that you can just write it off, but this goes into your risk assessment as being low risk materials for skin contacting. Like I said, FDA, this isn't an approved guidance document yet, but it is draft and been commented on. So it is current thinking from the FDA. This is the naughty list. I'm not going to get into why these are on the naughty list and things like that. Most of them are metals. If you want more of this, like I said, you can go to the webinar. But these are devices or materials that even if it's contacting the skin, you may want to do a little bit more of an assessment on the potential risk. Not saying you automatically are stuck doing full testing if it's on the naughty list. It just means you can't really apply those risk-based material assessments. Okay, so the other interesting part, and this comes straight out of ISO 1093-12. So this is table 1 out of -12. And the important part here that I want to talk about with changes is this little box right here, which is the extraction ratio has a plus or minus 10% kind of give grace period there to be able to help labs understand that if you're within plus or minus 10%, then you can compare the results from testing. So it gives us a little bit of room with surface area calculations, and when we do our surface area. I like to consider this concept the same with change. If you're plus or minus 10% of the total patient contacting surface area of your medical device that's changing, if you're less than 10% of it, then obviously the risk to the medical device impact to a biocompatibility test is really low, because over 90% of that extraction is going to be exactly the same. Where if you start to get over 10%, then you can start having an impact on your biocompatibility tests, and then you really start trying to, obviously the risks, not just the patient, but also to the testing itself, becomes a little bit more. If you're changing more than 50% of your patient contacting surface area, that's something you should really start to consider. Okay, so I'm going to be really high level on these, but these are some of the tools. Let's say you are changing more than 10%, your device is on the naughty, or your material's on the naughty list, or you're changing a portion that's in tissue or blood contacting. So what are some tools you have to really evaluate or mitigate the risk of that change? The first one is chemistry. And I think this is one of the most powerful things that we have with extractable leachables. A lot of people right now are frustrated with extractable leachables, specifically with the acceptance through like the FDA, and it's been creating some backlog within the testing CROs. But that being said, there are some chemical characterizations that can be used, some more easy and quick chemistry that can be used to help evaluate a change. These are things like FTIR or DSC, or even some GC or ICP-MS, portions of chemistry that we can be used that are not the full-blown extractable leachable, really expensive long-term types of tests. For example, if your supplier is telling you that they changed their polypropylene to a exact same polypropylene, maybe they just changed an additive or something, FTIR can help actually show that. So you run FTIR on your previous material and your new material and really see that. DSC is the same kind of concept. Or let's say you're changing a metal and you want to see how similar your new metal is to your old one. Maybe you're even changing types of stainless steel. ICP-MS will allow you to kind of really understand the type of leachable metals that leaching off of those metals. And then we can evaluate that from a toxicology standpoint. The other thing that a lot of people look at as a tool for this is cytotoxicity, which we'll talk about. And Cytotox is a great tool for changes. Just have to warn you, Cytotox is pretty sensitive. So if you fell, that doesn't mean that your change is now going to hurt somebody. But if you passed on your previous device and you're failing on your new device, it does mean we have to evaluate what's different, and then understand if it is a patient risk or not. So really high level, I want to make sure I give John time to talk about on his side. But this is kind of chemistry in a nutshell. When we look at chemistry, when you do a full extractable leachables, we're looking at all the organics that we can and all the inorganics. And this is because we don't really know what we're looking for. So we're trying to screen for everything that we can coming off a medical device. But with changes, we may not be needing to look for everything. We may know what's changing, and we can run some chemistry that's focused on the change itself, not necessarily on everything that could be there. So these are things like metals, like I talked about with ICP-MS, maybe we can focus there. They're also, with the organics, we can separate these out into VOCs, SVOCs, and non-NVOCs, which is small, so that's volatile, semi-volatile, and non-volatile. These are just the molecular weight of these compounds. The small ones are volatile, which is like alcohol. They evaporate really quickly, where your large, big molecules are non-volatiles, like lotions and greases. They stick around. So when you start looking at the change, we may be able to pick out what type of organic method to run based on the type of change. It doesn't make a lot of sense necessarily to run everything if we know what's changing and we can focus that chemistry on exactly what's changing. When we run this, we get a fingerprint from the device. This fingerprint helps us identify those compounds. And so that's how we can kind of compare. Let's say, for example, you ran chemistry before your change. Now we did extensive changing and we have to rerun chemistry. We can actually compare these two fingerprints side by side and really understand what changed or what impact the change may have. So we're looking at any compound that might be higher than the original or unique to the original device. So these are techniques we can run help mitigate the risk of a change. And this is a substantial change that you'll need chemistry to justify. The next one, like I said, is Cytotox. Cytotox is a very cheap, quick test. Here at Nelson Labs, you get a week to be able to get, a full report for Cytotox. There are some problems with cytotoxicity. For example, latex, rubber, silver, copper, dark inks, short curing times, adhesives. All those could cause cytotoxicity, but may not be clinically relevant. So for this, we call Cytotox your best friend and your worst enemy. Because it is very sensitive, very quick, very cheap, it's a great screening tool. But your worst enemy because it fails, and when it fails, it's not necessarily clinically relevant. It could be, but those investigations need to happen. So use this tool appropriately. Very high level, this is done in vitro on L929 cells, which are mouse fibroblast cells. We grow them up and put them on a plate, and we extract your device, and right here, we put that extract onto the cells, and then we just see how the cells respond. So a 0, 1, 2, 3, 4 based on cell death. 2 or less is considered passive. But the good thing here is that you can even if you have a device, an original device that had a cytotoxicity result but you know is safe, you can still run cytotox on your new device. We can run dilutions and find an endpoint of toxicity, kind of like a dose response curve, to be able to show that the new device has the same or better cytotoxicity response as the original device. So this is a 0. These are cells that are healthy and happy compared to this, which is a 4. As you can see, there's actually quite a big difference between those two. Where it gets hard is to score like a 1, 2, or 3. That takes some training and work. Just to give you an idea what they look like. This is my favorite picture movie. I show it every Friday night to my family. So this is something we like to see the cells growing, spreading, being happy. The happier those cells are, that means the less toxicity that's interacting with them. Like I said, they're very sensitive. They're basically bathing in your chemistry for 48 hours. So anything, or 72 hours depending. So anything that comes off your device is just sitting there and interacting with those cells. Okay, at the very end of your change, though, after you go through all the initial risk assessments and any testing you feel is necessary, please don't forget about making a report. We call them biological evaluation reports. You can call them risk assessments. You can call them whatever you want. But you should have something that talks about why you're doing this testing, or the risk evaluation you initially did, the risk that you identified, the testing you're doing and why to mitigate those risks, and then the results of those testing, why you conclude that the risk has been mitigated. Right? So there has to be some kind of story that's written to help you evaluate those changes, not just from what John's going to talk about here in a second, but also from a biocompatibility standpoint, really walking through that thought process so that when auditor or submission, they can follow your thought process and agree with it. So that's a very important part that a lot of people forget. They'll make a change, they'll just throw their ICP-MS results into the FDA and say, this is why it's good. And you're trusting that the reviewer can make those jumps, right? So we need to walk the reviewer through it ourselves. So that's the conclusion from a change that you're looking at. Now, I know I went kind of quick, but I want to make sure John gets some time to go through his thing. So I'm going to turn the time over to John, and there will be time for Q&A at the end. So if there are some questions that you guys have, please make sure you put them in there. But right now, John, go ahead and take it away.
Thank you, Thor. And folks, it's a great pleasure to have the opportunity to share the stage with Thor Rollins and Nelson Laboratories today. I can tell you that Thor and the team at Nelson has gotten me, well, I hope this sounds right, gotten me out of a jam or two on the topic of biocompatibility when making some changes many times in the past. So definitely a resource that you want in your corner when it comes to biocompatibility in general. And I guess another thing to remind folks too, yes, we are talking about the impact of design changes and biocompatibility today. But a lot of the things that we're covering are also applicable if you're just getting started as well. So keep that in mind. But I want to talk a little bit about some of the ability or things that you need to focus on from a documentation point of view. So, whoop, I got a little bit ahead of myself here. So, all right. So just like with any other change control process, if you're making the change of a material for whatever reason that you need to. Of course, you're going to need to go through all the steps that Thor described and shared with you all just a few moments ago. But there's some things that you need to do internally as well to document your decision-making process. So hopefully you have a robust change management practices in place. As you would expect with any other change that you're making, you're going to describe why you're making that change. You're going to provide a justification to explain that. Maybe you have to use a different because from a new vendor, or the material you were using is no longer available, or whatever the case may be. But you also need to think about the impact of these types of changes. What does this mean to your current inventory of product? What about product that's in the field and things of that nature? And you need to identify who needs to be involved in that change management review. So the intent here, as with anything, just about everything we do as medical device professionals, is we want to be able to demonstrate clear traceability and connect the dots, so to speak, so that when you make decisions, you're documenting the rationale for those decisions, and you're supporting those decisions with clear objective evidence, whether that be biocompatibility testing or some sort of material characterization, or whatever the case may be, that this is the evidence that you need to support your change in your decision making. When you make a design change to your product that impacts the materials, this is a design control thing. And you maybe already have gone through design and development, design control activities, have a complete design history file, and have already passed that threshold where you're already in the marketplace. Even if that is the case, there are still design control ramifications that you need to consider. My advice to you is your traceability matrix that shows the relationship, the flow of information from user needs through design inputs, outputs, verification and validation, and all those design reviews that you conduct along the way. This, in my opinion, and I think a lot of others will agree with this, is you need to take a total product lifecycle approach to your design history file. This should be a living entity. This should constantly be updated to reflect the current product and the state of that product on the market. So if you make a change to a material, you need to assess the impact from a design control perspective. You need to review the impacted design input requirements that could be affected by that particular material change. Of course, you need to update your design outputs accordingly, and you're going to need to do at least verification and probably some sort of validation activities as well, even if that is a rationale and an explanation as to why you do or don't need to do certain activities. But nonetheless, these are all things that need to be documented. And Thor talked about the reports and things that you might get from Nelson Labs whenever you conduct these design changes, material changes. Well, these are all things that should be contained within your design history file. These are all great examples of verification activities. And I want to heed he didn't state it as a warning, but I do want to heed the advice that Thor provided you. And I've made this mistake. It's very easy to get the reports, your biocompatibility reports from Nelson Labs, and just want to drop that into your 510K submission and call it a day. But when you do so, to Thor's point, you're assuming that reviewer has some expertise in biocompatibility and can draw the conclusions. But that's not a very good assumption. It's a poor assumption, because chances are, there might be somebody that has some biocompatibility expertise from the regulatory body that's reviewing your information. But don't make that assumption. You should summarize that. You should draw the conclusion based on the evidence that you're provided, and draft those summary reports and provide that as the objective evidence. But again, this is all information that should be contained within your traceability matrix and documented as part of your design history file. I guess the regulatory framework for this, and we're giving some citations to FDA 21 CFR Part 820, specifically 820.30 with respect to design changes. This is also well defined and very, very similar within ISO 13485. The good news for you when it comes to design and development types of activities and design changes, both FDA and ISO 13485 are very much in sync and alignment with one another. But the expectation is that you're defining your processes and procedures for when design changes happen and determining when design controls are impacted, when you need to validate and verify and review that type of information. Kind of a rule of thumb that I think is pretty common in the industry when you make a change, and this can be definitely material change, but just a change to a device in general. When you're making that change, kind of the generic questions that you should be asking yourself is, does this change have an impact on the form? Does it have an impact on the fit? Does it have an impact on the function? And if you answer yes to any of those, then that implies, oh, I should be doing something from a design control perspective, whatever that may be, whatever is appropriate and applicable to that type of change. And if the answer is no to all of those questions, that's totally conceivable. But make sure you're documenting a rationale and a justification and an explanation for why the answer is no. Just saying no or NA does not cut it in today's regulatory environment. So if you say no, just justify it, provide an explanation for why. Further, talking about changes, and specifically in this case, document changes, if you refer to the FDA 21 CFR 820.40, this is about documentation and record management. It gives some expectations with respect to changes to documents. So if you change a material, well, first and foremost, you should have some sort of material or I would expect you to have some sort of material specification or some sort of document or record that describes what that particular material is. You probably have a DMR, or you should have, you better have a DMR, device master record, that explains the materials, the components, the quantity, the size, the dimensions, all these things about your product as well. Well, if you're changing a material, something about your DMR is changing, something about your documentation is changing. So this is why having a robust change management practice is important. And again, I know we've cited CFR 820.40, but the ISO 13485 expectations and requirements when it comes to document changes are very much synonymous and in sync with what FDA defines as well. Speaking of EU MDR, Thor mentioned this a little bit earlier, but obviously that we're in a state in August of 2021 where EU MDR has been live for a few months now, and I know we're all trying to figure out and determine what this means in the grand scheme of things, how this is going to all play out. So I know we're all there right now, but the EU MDR puts a ton of emphasis on total lifecycle traceability. This is what I mentioned a moment ago. Treat your design history file, your design and development file, as living throughout that entire product lifecycle, and you need to demonstrate that full traceability. And this is one of those things that I think has evolved over time. I mean, I've been in the medical device industry since the late 90s, and during those first few years of my career, design history files were oftentimes archived the moment that we made that full transfer to manufacturing. And today that practice has changed, because you need to keep this as living, because a design history file is an important set of records that describe your product and shows what you did to demonstrate that your product is safe and effective. That it works, and the evidence that supports that. So, just keep those design history files up to date. That way you're always prepared. And, some of the things that we, with respect to Greenlight Guru and the medical device success platform that we have, this is designed specifically and exclusively for medical device companies. We've built the platform with FDA QSR, ISO 13485, EU MDR, and all those applicable regulatory requirements. Those are the framework by which the Greenlight Guru software platform is built. And we have recently introduced some functionality within the platform that allows or includes some AI and some machine learning within our intelligent document management system. And the advantage here is, maybe you have a material that you're using that is used in a number of products. Maybe it's referenced in a number of different documents or records, whatever the case may be. And if you make that change to that material, sometimes it can be challenging and very difficult to know everywhere and all the products where that particular material is used. Well, with the intelligent document management within the Greenlight Guru medical device success platform, you're able to get this intel right at your fingertips. The system will identify the applicable documents and records and things that could be impacted by this particular material change. Because sometimes you may want to change a material or something about a design, and it may seem like a really good idea, but if you don't know that impact, what other products or processes, et cetera, are potentially impacted by that, you could be making a decision in a vacuum. So the intelligent document management functionality gives you more intel and more knowledge about the impact of this particular change. And so if you know that this particular change is going to impact a dozen of your products and required updating 100 documents, you may decide maybe now is not the time to make that change. But nonetheless, it just gives you more information to make better decisions about your design changes. Mm-hmm. So last, I want to wrap things up, talk a little bit about the relationship between Greenlight Guru and Nelson Labs. We are partners, strategic partners working together. We understand the need for streamlining your product development activities, making sure that you are on the right path with respect to what is expected and required from a testing and a documentation standpoint. As I mentioned a moment ago, Nelson Labs, Thor, Rollins, I mean, these folks are biocompatibility experts. They're helping to write the guidances and standards that are impacting us as medical device companies. So it's a really great combination and really great partnership, and we're both here in your corner to help you get your products to market, keep them in the market so that they could continue to be safe and effective and help improve the quality of life. So as Thor mentioned, we want to make sure we have plenty of time for questions, and if I'm guessing correctly, there's probably quite a few questions that folks may have on the topic of biocompatibility and design changes and documenting those things. So fire away, and if you still have questions that haven't got them in, there's still time. We have a few minutes that we're going to be around to answer as many of those questions as we can in the time that's allowed.
Well, thank you, Thor and John, for your insights in this presentation today. Yes, we are seeing the questions starting to add up. So we've received a lot, and you've given us a lot to think about. So I'm just going to start asking some questions, and I'll let either of you take it. And if one of you want to take it and the other one wants to chime in, that's perfectly acceptable. So I'm just going to start asking some questions to you. So I'm going to put this first one out here. The first one is, when doing an adoption assessment, what is the relevance of comparing surface area between the existing and candidate device? Is the plus minus 10% a generally acceptable value to consider, unacceptable, or acceptable for adoption as well?
Yeah, I'll take this. And John, if you have thoughts, always love hearing your thoughts. So yeah, it is acceptable. So, but the thing about adoption, if you're adopting a product or if you're looking to do a family grouping from a biocompatibility standpoint, the thing to think about is from a testing or just anything from a biocompatibility, it's about materials surface area ratios when we test. So a lot of times you start looking at, okay, can I get a family grouping or adopt a product into something I've already tested that has very similar materials and processes? So if you go back to that initial risk assessment, you look at the materials and the processes. If you can group them together in those, then surface area does become the question. But if it's all the exact same materials and processes, just different sizes, for example, for me, it doesn't matter because I'm going to keep my surface area ratio to volume consistent when I test. So if you send me something that's 1,000 centimeters squared or 100 centimeters squared, I'm going to test them the same. I'm just going to add more volume to the 1,000 than I do the 100. So really, there is no difference between there. The real key comes to when you may have products that have maybe a little bit different types of materials or different sizes of some material versus others. Then coming up with a representative device that can do that family, and then do some kind of adoption if you have a new product into that. The plus or minus 10% does help when you start looking at the differences between those materials. So yeah, it does play a role, but there's a lot more to think about with the product adoption than just that 10%. John, I don't know if you have anything to add there.
Just a brief comment. I think in my experience, a lot of companies have staff who have gained quite a bit of expertise on the topic of biocompatibility, and I think it's a smart move, especially if you have products that are patient contacting in some way, shape, or another. Encourage some of your staff to get some formal training on this particular topic. But even so, even though you might be within those thresholds, I found that having an expert such as Nelson Labs in your corner that helps draft the assessment for you will carry a lot more weight with regulatory than if I were to do that assessment by myself.
Great, thank you, Thor and John. Here's our next question. Is a biological evaluation plan a requirement of ISO 10993-1 or just a good idea?
Yeah, John, I'd also love to hear your thoughts on this one. So for me, okay, a risk-based approach is a requirement. So if you look at this, read 10993-1, evaluating the risk of your medical device based on the materials and processes, and then mitigating that with the testing that you think is sufficient is required. Some people will just do the testing like a checkbox, and the FDA right now still accepts that, but Europe does not. Europe will want you to have some kind of assessment done to justify why you tested, right? So even if you just did all the testing, they're going to say, well, you did a lot of animal testing. Why did you do a lot of animal testing? What was the risk that you were trying to mitigate? So a BEP is not officially required, especially for the FDA. Like I said, if you just did all the testing checkbox, they tend to be okay with that. But for me, a BEP is almost required. I mean, especially unless you have something that's like a skin contacting device that that you're just going to do the big 3 on and send it in, that might not be required. But anything that does any kind of toxicity, any kind of prolonged genotox, carcinogenicity need for evaluation, I don't know how you, like John said it good too, I don't know how you put together your thought process in a story form without a biological evaluation plan and a biological evaluation report. I just think they're essential if you start to have those conversations. So they're not technically required as far as a document, but that kind of risk assessment is required for Europe, and I think it's almost required for most devices for FDA. John?
Yeah, it's just that, I mean, it's a best business practice, and as a medical device company, I think we are all looking for those best business practices. It's a communication tool. I mean, maybe in the moment in time, you may think, oh, well, this biological evaluation plan doesn't really help us all that much, doesn't really add a lot of value. And, you might be right. But what happens 3 years from today? And, somebody, some of the people who were involved in the decision today aren't going to be here in 3 years, or people forget things and it's not documented and written down. You may not understand, the people then may not understand why the decisions that were made. So a biological evaluation plan becomes a wonderful record to help explain the rationale, the thought processes, and the approach that you took. So I think it is, even though it may not be required by all regulatory bodies, we shouldn't just do things because regulatory bodies require them. This is just the best practice.
Actually, John, I want to add, you said something perfect there. With this new MDR, us going back and trying to help customers do gap analysis to meet the current standard, and they've done testing that we don't think is needed, and we ask them, why'd you guys do this? And all the time they'll say, well, either I wasn't there or we're not sure. We just always, like, there's been no thought or documented thought on why we're doing what we're doing. So having something like that to help you in the future, because you're going to forget too in a couple of years, is very important. John makes a good point.
Great. Let's move on to our next question. So, is the biocompatibility requirement the same for devices that are in contact with the skin all day as glasses that the devices the patients only touch a few minutes per day?
So it depends, which is the best regulatory and biocomp answer that exists. If you look at the 10993-1, there is still a chart in there that helps you. It's in an annex, informative annex, but it helps you evaluate what biological endpoints are needed. For something that touches intact skin only, it really doesn't matter much from a biological risk perspective. If it's on the touching your nose for like a minute a day, or for just a couple of minutes, to something that's always in contact with your skin, you're pretty much looking at the same endpoints. Where it becomes different is once you start going to breached or compromised, or something that touches the tissue or mucosal membrane. Then the dwell time becomes important, and then you just follow the risk. So it does depend on the type of body contact. Sometimes it doesn't matter. Sometimes it matters a lot. It just depends on where the device is contacting.
Great. Let's go on to the next question here. Chemical characterization seems to be the new gold standard to justify out of long-term toxicity. Do you foresee countries like Japan accepting this in the future?
Oh, so Japan's accepting it now. Now you have to be careful with that because you have to kind of make sure that you're doing it for them. But a lot of our E&L studies have gone to Japan right now, and we've been successful there. Same with China, Korea. Honestly, the only, shouldn't say this way, but the only trouble regulatory-wise we've had with current E&L is FDA, and that's just because their bar is so much higher than anywhere else, which I understand why, because once you understand chemistry more, it's easier to see the holes in chemistry than it is the animal test, because we know more, right? So you got a lot more knowledge, and when you have a lot more knowledge, you see a lot more. With the animal test, we don't have the knowledge, and so we don't see the gaps, and so we feel more confident in that. So the FDA has really elevated the bar for E&L. It is the gold standard. The reason why is because it's very hard to evaluate true chronic and especially carcinogenicity risks from a medical device without using good E&L. There's a potential to do animal tests for carcinogenicity, but it's so expensive and long, and it's really not that great of data. Where chemistry allows us to get those endpoints now more cost effectively. That being said, like I said, we're in the middle of a revolution, honestly, with E&L. Trying to hit the requirements and make sure that we're not missing anything because of the amount of weight that we're putting on E&L, like you said, toxicity, carcinogenicity, genotoxicity, these endpoints. But yeah, we're seeing it accepted right now. At least I can only speak from Nelson Labs' point of view. In Japan, in Korea, in China, in Europe especially, and in FDA if it's done correctly. John, I don't know if you have anything that you want there.
I do not.
Great. We have a time for just a couple more questions. Thor, I do have, I think this was under your slide. If you could go back to the naughty versus nice materials list, they would like to take some screenshots. And here's our next question. ISO 10993-1 indicates biocompatibility should be verified throughout shelf life. Where does industry regulatory bodies stand on that, and what's the best way to perform it?
Oh, wow. We could do a whole 2-hour webinar on this. So, yes, the new standard requires biocompatibility to be evaluated throughout the product lifecycle, which is shelf life. We're getting this question from notified bodies with the new MDR. Okay, so we are getting this question now, and we are addressing it. The truth of it, though, is the way to really address it from a scientific standpoint is not best from a timing and cost perspective. Because really what you should do is perform extractable leachables on a brand new device, real-time age that device, and then do extractable leachables on the end product and compare them. You could do accelerated aging, but there's some discussion about the true leachability differences, so you could do all three. Honestly, though, we don't have anybody doing that just because of time. So how we're addressing it is materials assessment in our biological evaluation plan, right? So we look at your materials, we look at their stability. If we feel like there's a risk of stability, then we will try to run some biocompatibility on aged samples like Cytotox or some simple chemistry. And so we are addressing that today. The reason we're addressing it today is because we're starting to see routinely feedback from notified bodies on, well, how do you know your biocompatibility hasn't changed during your shelf life? And the only way to do that is to actually, have a justification of why. For example, if it's a piece of titanium, you're pretty confident that after sitting 3 years, it's not going to get more toxic. But some of these polymers, you really can't say that. You could have cross-linking that breaks down, or things that start to bloom to the top that you wouldn't have at the beginning. So the only way to really address that is do some if you feel like there's a risk, do some Cytotox on an aged sample, or do some comparison chemistry-wise on some aged sample. But what we write our BEPs, we take that in consideration by doing a material research first, and then recommending testing to address that if we feel like there's a risk. John, I don't know if you guys are seeing that. I know that you talked about shelf life or like life cycles. I don't know if you have anything you wanted to add there.
Well, yeah, I think it's, yes, you need to address the biocompatibility through the shelf life. And, a lot of single-use disposable products have a target of 3 years or maybe even 5 years from a shelf life perspective. And a lot of times when you get those products through the initial regulatory clearance or approval or whatever the case may be, a lot of times you are relying on some sort of accelerated aging to demonstrate that. But because you don't have that luxury of waiting 3 years to introduce your product. In addition to the biocompatibility concerns, there's also an expectation that you're able to demonstrate the product performs through its entire shelf life too. So my advice to you, if this applies, if you have products where biocompatibility is something to address, is that you factor this into your design transfer planning and your product planning, that you're holding back quantities of product so that they can sit on a shelf in your storage facilities, wherever that may be, and you test them at different periods of time, real time, both from a biocompatibility perspective, but also from a performance and a sterilization perspective. These are all factors that we need to be cognizant of.
Yeah, and I also just wanted to add, I totally agree with John. In fact, we have customers who do that, and I think it's great data. The FDA is also concerned about this from our experience, but it's more based on the risk of the device, right? So if you have materials like a biodegradable material or a biologic or a tissue, that's when the FDA starts getting concerned a lot about biocomp. They tend not to have as much concern about a piece of polymer or a piece of metal. But John's point is 100% correct.
Well, great. Thank you, Thor and John. We are out of time. We'd like to thank you for that great presentation and all the questions that you were able to answer. I'd like to thank Thor and John for their insights, and thank you for this presentation today. I'd like to thank you, the viewer, for attending this session. We hope that you found it valuable and informative. At the end of the webinar, please take time to fill out our brief survey that as we value your feedback. And we will try to get to as the remaining questions as well will be sent to our presenters for them to be able to respond as well. And next week, you will be able to find this webinar on the Nelson Labs website for reviewing again. Again, we thank you for attending today, and we hope you have a great day.